Hey, what's up everybody, and welcome back to the Sanctuary. I'm Professor C, and we'll be talking about anatomy and physiology again today. Specifically, we'll be looking at our third group of organic molecules, the proteins. So, let's get into it. Okay, let's first talk about the elemental components of proteins. John, no surprise that we have CHO. We've seen that in both the carbs and the lipids. And except for things like, phospholipids and slight adjustment to the basic lipid structure, we had CHO predominantly, only. Now we're going to add some N. Nitrogen will be in all proteins. Sometimes S, sometimes P, so we see a lot more variety of elements that make up proteins. We've seen this a few times already, but I'll restate it. If you want to draw a protein in its simplest form, it's just a polymer of amino acids linked by peptide bonds. The key here that we haven't seen is the word folded. So what we're going to do is take this necklace of amino acids that are linked by peptide bonds and fold them. And you can see in the background picture there all those red curls and blue curls and these twists and turns. There's going to be different structures that we can fold these polymers into that will give it a nice three-dimensional shape that will allow it to have a very, very, very specific function. And there are many functions of proteins in cells in bodies, so we can't talk about all of them, but we will probably talk about 5 big ones after we get past the concept of folding a protein. So there are going to be 4 levels of protein folding. And the first one, sometimes you can just shorthand the word primary as 1 with a little circle above it. That means primary. So the first level of protein folding is actually not folded at all, which is strange. But the string of unfolded amino acids, and you see them there, each one of these balls is an amino acid, and the bonds that hold them together is called a peptide bond, just like we went over in the last slide. Now, we will see our friend carboxyl at one end, but at the other end of this tail, we see something new, and it's an NH2 group. And this is called an amine or an amino end. So at one end of your protein structure, you'll have a carboxyl, and the other end you'll have an amino end with a nitrogen in it. Now at this point, where you only have a string of amino acids linked by peptide bonds, you probably would get away with calling that a polypeptide more than a protein. And we'll talk about why in a minute, but the quick answer is you have to fold up that polypeptide more than just this unfolded primary structure in order to call it a real deal protein. So usually a short unfolded chain of amino acids is called a polypeptide. So there's the first way to fold a protein. Well, take that string of amino acids, and you can see that right here in this part of the picture. There's a string of amino acids, like the necklace. You can't see the peptide bonds, but they're buried in there. And then start to twist that string, and twist it and twist it and twist it until you get this coil. And if you can do that, you've done one of the 2 secondary folds to a protein. That's one of them. It's called an alpha helix. A coil, a helix. So if you can twist and twist and twist this string of amino acids, the primary structure, you can twist it into a coil. It's called an alpha helix. There's another thing you can do too, is you can fold it like a sheet of paper that's folded into pleats. And this is called a beta pleated sheet. So it says down here, very long chains of amino acids twisted into coils or folded into sheets. And then here's our ... that we've seen at least one time in the past. And we know that a ... tells us that's a hydrogen bond or an H bond, a very weak bond holding this spring or pleated sheet structure together very loosely. If you want to go further, you can. You can get into the third level, tertiary folding. And all you have to do now is take multiple alpha helices. You see one back here. And then here's a pleated sheet running along there. Here's a pleated sheet here. Here's a pleated sheet here. Here's another alpha helix. Take a lot of the springs and a lot of the sheets and tie them all together in a very, very long structure. Same thing up here. Multiple alpha helices, multiple beta pleated sheets linked in series, folded up into a tight globular protein, held together by intramolecular H bonds and other types of bonds like a disulfide bridge, which is an SS bond. So this is a tertiarily folded protein. Not only a string of amino acids that's been twisted and folded, but then a series of twists and folds and twists and folds, and then folded up again into a 3D shape. This is tertiary. Well, what else can we do then? What is possibly another level of folding? Well, simply take the globular protein that we made in the third level and add them together. This is the fourth level, or the quaternary level of protein folding. So for instance, take this blue protein, which is a third level folded protein. I can see multiple sheets, multiple helices. Here too is another globular protein, and here too another globular protein, and here's a fourth one. So in this drawing, we not only have 2 globular proteins, but 4 of them. And this guy is called hemoglobin, something you've probably heard of before. So hemoglobin is made of 4 three-dimensionally folded globular proteins. 2 are called alpha subunits and 2 are called betas. We're not going to really worry about that part here because it confuses us with the alpha and beta pleated sheets. So we'll save that for the blood unit far in our future. But this is 4th level folding. Take multiple globular proteins and link them together, creating a single functional unit. So let's go over some big functions. I said there are many, many functions of proteins, but you can kind of boil them down into large categories like we're really good at doing here in our study sessions. So let's change the pen up a little bit. Function number 1, proteins act as receptors. Again, the picture is a little too much for us, but I can see in the structure a lot of curls. I can see a lot of alpha helices. I can see some beta pleated sheets if I look hard enough. And I can kind of see that this is in some kind of boundary between outside of a cell and inside of a cell. So we're looking at receptors here. We would draw these in our pictures as giant Y-shaped structures poking out of the cell membrane. But we've heard the story of receptors before in homeostatic control mechanisms. A lot of receptors are proteins. Function number 2 of a protein, proteins act as transporters. We will definitely talk a lot more about this later, but the point is, if I have a membrane, and you can see it right here, there's our cell membrane. I can see some little structures we saw already. These little balls of 2 sticks, and that's phospholipids, isn't it? So there's a cell membrane, and you can see these proteins spanning the membrane and letting things come across the border, so to speak. So proteins act as transporters, carrying things across that very almost impenetrable layer of phospholipids. Okay, a third major class functionally for proteins is proteins act as enzymes. And the way it's written here, primary component of enzymes is protein. Sometimes an enzyme is completely protein and has no other component. One of the tricks we can learn is ACE. 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 ACE is our word for enzyme. And we mentioned enzymes when we talked about rates of reactions at the end of our chemical reactions unit. And we talked about how enzymes can speed up reactions or make them more efficient for sure, without being used or destroyed by that reaction. Fourth big function for proteins is that they act as antibodies. These Y-shaped structures here, not receptors in this case, but they look like they're going after this thingamajigger here, trying to take him down. So these Y-shaped antibodies are, we all know about these, they're for defense, right? They're for the immune system. So if you want to study immunology, at some point you'll have to get into the very specific mechanisms of how antibodies work, and you'll find out when you do that they are made of protein chains linked together. The fifth major class of protein is just that it serves as a structural component of many different tissues and many different cells. Think about your hair, your fingernails, structures within your skin, to keep it waterproofed, to keep it, a little bit stretchy. Think about proteins you will find in the heart, like actin and myosin, that help muscle in your heart contract, or muscles in your skeletal muscles move back and forth. And I have noticed this as well. I know that if I see ACE at the end of a word, that tells me it's an enzyme and probably a protein. But I've seen this root IN a lot here. Hemoglobin, keratin, elastin, a lot of them. N sometimes tells you that you're looking at a protein because it ends in N also. OS for sugar, ACE for enzyme protein, and N sometimes for just a normal protein. To denature an enzyme, or a protein in this case, regardless if it's an enzyme or not, is an unraveling. So remember, if you want a protein to function correctly, you need to have it folded up correctly and have a good three-dimensional form so it can do its function, right? Structure begets function, going all the way back to the beginning of the course. However, if you have extreme environmental changes like extreme temperature disruption, or extreme pH changes, or extreme pressure changes, you can change the shape of the protein, even unravel it forever, even irreversibly. You can unravel it. This is a denatured protein. You've lost your structure, so you've lost your function. And this is the problem with denaturing proteins, is they lose their function. As usual, thanks for watching the video. If you want, check out the other videos in the series, and I'll see you next time. Bye-bye.